The biology revision video for photosynthesis, and today we're going to be looking at all the stages of photosynthesis. You may want to break this video into several parts. You need to know the overall equation for photosynthesis, which is water plus carbon dioxide goes to oxygen and glucose, and that is obviously using sunlight, which provides the energy for photosynthesis. And that is important. It's converting the energy from sunlight into chemical energy, which is stored in originally as glucose and then other carbon compounds such as protein, lipids and other carbohydrates. So for example, the glucose can be converted to cellulose. This is obviously important because it provides the energy in most food chains. Now, this reaction starts by doing something called photolysis, which is where we take a water molecule and the energy in light, and that's what the energy is used for, one of the reasons in photosynthesis, splits that water molecule into two hydrogen ions, two electrons, and half an O2 molecule. So if there's a balance, you'd actually have two water there to make 4 lots of that, 4 lots of that, and one O2 molecule. And this then gives these electrons here, which can then be used in this reaction, as you will see as we move forward. So photosynthesis is effectively the opposite reaction of aerobic respiration, and this helps with the carbon cycle. So if you think about it, the products of photosynthesis are the reactants of respiration, and the products of respiration are the reactants of photosynthesis. So there is a cycle involving these organisms releasing carbon dioxide and absorbing carbon dioxide. Photosynthesis occurs because pigments absorb light energy. So the job of pigment is to absorb light energy. And chlorophyll is the most commonly one that we will talk about, is a green pigment with magnesium in its center. And chlorophyll is found in the thylakoids of the chloroplasts, and it absorbs light energy apart from in the wavelength of green. These are attached to proteins which help them funnel the energy to where they need to be, and that is called a photo. So plants don't just have chlorophyll though, they have lots of pigments, and therefore they can absorb light at lots of different wavelengths, maximizing the efficiency of photosynthesis. So in higher plants, they're generally split into two groups, chlorophylls and carotenes. And there is something called the absorption and action spectrums. So the absorption spectrum is a graph that shows you the absorption of different wavelengths. So if we look at chlorophyll A, this line here, it absorbs high in blue light, it drops right down in sort of yellowy green light, and then it jumps back up a bit in red light. And you'd be expected to be able to draw this sort of curve here to show your knowledge of the overall area. So you'd be expected to know in the middle about 500 to 600, that's where you get the lowest absorption of light from chlorophyll, because it reflects green wavelengths of light. Now, for HL students only, you need to know about photosystems. So pigments are, as I said earlier, are attached to proteins in something called light harvesting clusters known as photosystems. There might be up to 30 accessory pigments which absorb all different colours of light, and one primary pigment here in the centre. Accessory pigments can be things like carotenoids, xanthophyll, chlorophyll B, or even different forms of chlorophyll A. And carotenoids you most common with carrots and oranges, which is why they look orange, because they reflect the orange light of the spectrum. And that's why we see them like that. They are different to photosynthesis, to chlorophyll, because they don't have any porphyrin. They're not directly involved in light-dependent reaction, but they pass their energy to chlorophyll A. So what we're saying is all these different pigments are in a protein or attached to a protein here. They capture the light from the sun, and the energy or the electrons that they have are energized and get passed from pigment to pigment down to this primary pigment center in the center. So photosystem 1 absorbs light best at 700, and the second photosystem 680. Each pigment absorbs a specific wavelength of light. This excites one electron in an atom to become excited and jump to a higher energy level. The light energy can then be re-emitted later on if the electron drops down to its original level, and this is called fluorescence. It explains why certain materials and chemicals will grow. And here you see a different way of showing it. The light energy gets absorbed by, in this case, they say a chlorophyll B molecule. The energy then gets passed on to other molecules and eventually gets to the primary center, which is. So it says here, light excites an electron in one atom to become excited, and as it gets passed along, the energy level drops as it moves along, and it all happens until it reaches. In the core complex, there is special chlorophyll molecules that then pass on excited electrons to the carrier proteins. We might want to look and investigate the chemicals, the pigments that are involved in photosynthesis. So we can do this using chromatography, which is used to separate substances in a mixture, and we can actually identify them by. And the principle of chromatography is that effectively you get your pigments, so that would be on that spot there, okay, and you put a solvent at the bottom of the paper. The solvent runs up the paper, that's called the mobile phase, and if the pigment is soluble in that solvent, it will be carried up the paper. Depending on what the pigment is, it will be carried different lengths because of its different solubility, and we can use where it is deposited to calculate the RF. You might have to do is calculate the RF values of different pigments in leaves, and it's important to know that leaves contain multiple pigments absorbing different wavelengths of light because that increases the range of wavelengths that the leaf can absorb. And in addition, photosynthetic leaves will have other pigments with essential roles such as UV protection, so it's not all just about absorbing sunlight. Different species will have different compositions. For the practical, you might have to calculate the RF value. So what you do is you leave the practical running until the solvent hits almost the top of the page, and then you can work out the distance from the line of origin to the compound or component, and the distance from the line of origin to the solvent front. And if you do that one divided by the other, you'll get an answer which should always be below 1. Okay, so there is your RF. That identify with known RF values and calculate or identify what chemical you have in your Let's look at how the process works then, which I believe is just HL, but you need to double check that yourself. There are 2 reactions in photosynthesis: the light dependent stage and or reaction, and the light independent stage. The light dependent stage happens in the thylakoid membranes. Remember, this is a chloroplast, not a cell, so in these membranes here. And the light independent reaction, often called the Kelvin cycle, happens in the stroma of the mitochondria of the chloroplasts. Inputs of the light dependent reaction are water, light, and something called oxidized NADP. And the outputs, the products, are ATP, which we will later use up, oxygen, which diffuses out into the atmosphere, and reduced NADP. The 2 products, ATP and reduced NADP, become useful in the final stage of photosynthesis, which we'll talk about after. There are actually 2 parts to the first reaction, the non-cyclic and cyclic photophosphorylation. So the main one we'll talk about is the first bit, non-cyclic photophosphorylation. And effectively, this shows you the whole reaction. So we start off in what we call photosystem 2. Photosystem 1 is over here. They named it 1 over there and 2 over here because of the order they were discovered. So in photosystem 2, light from the sun is absorbed and passed down, funneled down to chlorophyll A. The most absorbed amount of light is P680, so this is called P680, photosystem P680. The electrons in the magnesium in the middle of the chloroplast, in the chlorophyll, are excited. So the electrons get so excited they leave the chlorophyll, we'll get it right eventually, and they get passed into a series of electron carriers, which pass the electrons along. As they do this, hydrogens are pumped from the stroma, using the energy, into the thylakoid space, which is inside the thylakoid in this area here. These hydrogens build up, and we'll come back, well, we'll do that now actually, and they want to move back across via diffusion across the membrane, but they cannot get through this membrane. So the only way the hydrogens can get back through is via this enzyme here, which is ATP synthase, which you will recognize if you've talked about respiration. That ATP synthase effectively then gets something called a proton motive force when the protons move through, and it pushes the ATP synthase changing its shape, which forces phosphate and ADP together to make ATP. That ATP will go to the next stage of photosynthesis. The ADP has come from that other stage in photosynthesis. So we called it the light independent reaction. Okay, if we continue with the electron story, so the electrons moved down here to the second photosystem, okay, and the electrons got more sunlight absorbed in, get excited again, and they then move through another series of electron carriers or cytochromes, and they then are passed on to something called NADP, which is oxidized, reducing the NADP, making it reduced, and the NADP also accepts some of the hydrogens as well to make it reduced NADP. That NADP also goes to the light dependent reaction, and this has come from that reaction as well. So the absorption of light causes chlorophyll to become excited, and 680 is a powerful reducing agent and regains those electrons from water. So the reason we talked about water at the start of photolysis was the electrons released from the water replace the electrons that are excited from photosystem 2. There is something called an oxygen evolving complex on the interior surface of the thylakoid from photosystem 2, and it binds two water molecules and splits them from the energy, using the energy from light. So that's where that happens, and this is the equation showing you two water molecules breaks down into 4 hydrogens, 4 electrons, and oxygen, which diffuses out into the excited electrons then pass from photosystem 2 to the first. I called it an electron carrier earlier, or cytochrome, but its proper name is plastoquinone. It accepts 2 electrons to become plastoquinol, and then these electrons are passed down through several complexes to transfer energy to another complex called plastocyanin. So these electron carriers have different names, which you need to be aware of. Electrons are then passed along the to photosystem 1, and here they have a lower energy due to the fact they've been pumping hydrogens out. So then they gain some more energy from the sunlight, and then they are passed to reduced NADP reductase, which is the final electron carrier, which reduces NADP. So here's another picture that shows you it. So again, we've just talked our way through this. We have the water molecule here, which is split into oxygen and hydrogen and electrons. Those electrons replace the electrons that have lost here because they're excited, and they move into the electron carriers of photoquinone. And then you go through the electron carriers, they lose energy as they pass through. Hydrogens are pumped through from the stroma to the thylakoid space. In photosystem 2, more energy excites them, so they move to NADP reductase, which reduces the NADP. And the hydrogens pumped through go through the ATP synthase, generating ATP, and then are attached or reduce the NADP, which goes to the final stage. You must be able to talk yourself through that. That is called the non-cyclic photophosphorylation. And that's another method I've just shown you. So the water split up, the electrons go into photosystem 2, electrons get excited because of sunlight, pass through the electron carriers. Electron photosystem 1 get excited due to sunlight, pass through electron carriers, reducing the NADP and adding hydrogen to it to make reduced NADP. There is another process, which if you look at this version, you can see here with this dashed line, ATP can be made directly from this process, and that is called cyclic photophosphorylation. And basically, some of the electrons that are excited in photosystem 1 pass out of photosystem 1 but go down a different transport chain to go back to photosystem 1. So they replenish themselves and they generate some ATP doing this. And this ATP is believed to help open the stomata during when needed by opening the in the thylakoids, which you need to be aware of. Cyanobacteria have thylakoids that are actually attached to the plasma membrane, so more simplistic. Eukaryotic algae plants have two types of bacteria, of thylakoids, sorry. So there are disc-shaped thylakoids arranged in stacks called grana, which you'll be more familiar with. That's where photosystem 2 and the cytochrome B6 complexes are found. And then between the grana, there are what we call intergrana lamellae, which are the connections between the grana, and that's where photosystem 1 and ATP is found. Thylakoids are systems because they're interacting components individually could not function. Okay, and we just need to be aware of what they are again, so let's just summarize it up one last time. So you need to know that membranes separate the fluid of the lumen and of the thylakoid and the stroma, so the proton gradient is maintained. ATP synthase uses proton gradient to make ATP. The oxygen evolving complex in photosystem 2 splits water by photolysis and provides electron supply. Photosystem 2 itself absorbs light energy and passes it to plastoquinone. Plastoquinone and cytochrome B6F use energy from excited electrons to pump hydrogens to the thylakoid lumen, and plastocyanin transfers electrons from cytochrome B6F to photosystem 1. So you can see this is a system, because a system is a series of complex interactions working together. And also the last one, sorry, I should have said, photosystem 1 absorbs light energy to excite electrons, and these are used to reduce NADP on the stroma side of the thylakoid membrane. So once we've made this reduced NADP, and once we've made this ATP, what do we do? The reaction requires the use of products from the previous stage, and if light therefore is not available, this reaction would stop. So even though it's called the light independent stage, it doesn't rely on products from the previous reaction, but it doesn't use light directly. This is occurring in the stroma, and if we talk through it, we start with ribulose biphosphate, a 5-carbon compound. That then combines with, which you can't see here very well, carbon dioxide. It is fixed, the carbon dioxide is into ribulose biphosphate using the enzyme Rubisco. That makes a very quick, very temporary 6-carbon compound that's unstable and breaks down into 2 3-carbon glycerate-free phosphates. Glycerate-free phosphate then, using the products from the light-dependent reaction, gets reduced into TP triose phosphate. And that uses the reduced NADP, so that is oxidized, giving its hydrogens and electrons to GP, and energy is also utilized to convert that GP, reduce it into TP. Now that TP can be used to then make hexosugars such as glucose, which can be further then converted into things like starch, cellulose, or added to other molecules to make glycerol and lipids. But most of the time the TP is actually just regenerated using energy, again from the first light dependent stage, back into ribulose biphosphate, and the cycle goes round again. In terms of products, the 2 main products are triose phosphate, which is used to make glucose, and more ribulose biphosphate. But some of the GP is also converted into amino acids and fatty acids. Now, in terms of numbers, this cycle has to happen 6 times to create 1 glucose molecule. So out of every 12 triose molecules, remember there's 2 per cycle, 2 of those move on to make glucose. 10 of them are used to just reproduce ribulose biphosphate. Of the Kelvin cycle, a light independent reaction, are hexosugars, and as I said, they are made by joining up 2 triose phosphate molecules. They can be then further converted into other hexosugars as a result. These products are, so as I said, different carbohydrates. You can get lipids from triose phosphate and combine with fatty acids from the glycerate-free phosphate. So you can get a mixture of glycerol and fatty acids, and amino acids can be made direct from the glycerate-free phosphate as well. Evidence that someone created this was from someone called Calvin or Melvin Calvin, and what he did was he grew algae in nutrients, and then so they would grow, and after a certain amount he injected that algae with C14, so radioactive carbon, so we can actually monitor where the radiation goes. And then what he did is over different times he would drop the algae into methanol, which was hot, to kill the algae. He then analysed the chemicals produced, and over different time frame he was able to see what was produced. So when he did this, he first of all found at 0 seconds carbon dioxide was where all the radioactive carbon was. 5 seconds it would be glycerate-free phosphate. 10 seconds he identified it as glycerate triose phosphate. 15 seconds glycerate-free triose phosphate and glucose, and then he identified ribulose biphosphate as well, showing the order of the cycle from where the carbon dioxide would go that I've just spoken you. So there you go, so you see it here. So if you added the carbon dioxide, the first thing you would expect to see, which he got, was glycerate, sorry my pen's not working, glycerate-free phosphate, well he got that. Next thing, triose phosphate, well he got that. Next thing he got some glucose to show you how quickly that was made. And the last thing, ribulose biphosphate. So the order that we know it is from this reaction practical that he did. He could also radioactively trace it to look at different things in the light and dark, which allowed him to identify the need for the reduced NADP and ATP. So he found that glycerate-free phosphate levels increased once he added radioactive carbon dioxide in the light, because you would expect, but then when he took the light away, the levels increased even further, showing you needed whereas the ribulose biphosphate levels dropped, showing you needed that light to convert glycerate-free phosphate to triose phosphate and then to ribulose biphosphate. That link both reactions together, sorry, limiting factors that link both reactions in photosynthesis together, and you need to be able to link the two together to explain the impact of these limiting factors. So remember, the key thing is the products of the light-dependent stage, ATP and reduced NAD, are needed for the light-independent stage, or the Kelvin cycle. So if we run out of light, it will have a big impact, because we will not have this, we will not have this, and will not have this. And therefore, the reaction effectively without light is almost stopped there, because you cannot make any more TP without the light and the products from the light stage. So light intensity is a good limiting factor to talk about. So if you look here, we have light intensity on the graph, and light therefore is a limiting factor here. At point X though, something else must become a limiting factor, so it's either carbon dioxide or temperature at that point. Here you see a graph. Imagine this bit's joined together to make it easier. Light intensity is a limiting factor here, but once it's leveled off, it must be due to difference in carbon dioxide that there is difference in those graphs. So at that point, carbon dioxide is the limiting factor. Here, a different graph, you can see you've got a mixture of things here. So you have this thing here at the compensation point. That is the point where photosynthesis equals respiration. This is light intensity. So up until this point here, light intensity is the limiting factor, but then it levels off. Even if I increase my light intensity, this stays the same. So it must be a different factor, and it tells us it is carbon dioxide. So this graph here, you need to be able to explain how the levels of TP, ρBP, and GP change in the light and when they go into dark. So this goes back to what I was. So the level of GP is relatively low during light, and that is because the GP is converted using the products of light-dependent stage into TP, which also explains why the TP levels and then the ribulose biphosphate levels are high. But in the dark, these sources of energy, this is gone and this is gone. So the product stops there. The reaction stops there. So your GP levels go up and the TP and ρBP levels go down. There's an explanation for you. You can pause and read it if you would like. dioxide. Carbon dioxide is accepted by Rubisco, the enzyme, which converts RuBP into GP. So this is higher levels of carbon dioxide, this is lower levels. If we've got high levels, then you're going to have lots of GP being made, lots of TP being made. Ribulose biphosphate will be converted into GP, so you'll have low levels of that. But if the carbon dioxide levels drop, your ribulose biphosphate will no longer be accepting it, and therefore its levels